In this presentation, we'll be discussing serotonin, some of its biochemical processes, and its importance in brain chemistry. Despite being famously known for the role it plays as a neurotransmitter, where alongside dopamine, oxytocin, and endorphins, these have been labeled as the chemicals of happiness, and the neuromodulation of mood, appetite, and sleep-wake cycles, the bulk, from 90% of serotonin, is actually found in the intestine, inside enterochromaffin cells, which are specialized neuroendocrine cells that regulate intestinal movements. Particularly through neural signaling in the enteric nervous system, it causes peristaltic constrictions and reflexes of the gut. Serotonin has also been related to central nervous system cognition, where it's been associated with learning and working memory. The electrically excitable nerve cells called neurons communicate with each other through synaptic transmissions. Electrochemical waves called action potentials travel down the axon, which are elongated fibers that project over the adjacent neurons, what creates a quick polarization of this membrane. As this action potential reaches the dendrites of other cells, it provokes the release of neurotransmitters. The neurotransmitters are kept in small sacs called synaptic vesicles and are released into the synaptic cleft, the region in between two neurons, where it binds to the postsynaptic receptor and can either provoke further action potentials, exciting the neuron, or stop the electrical cascade, inhibiting the neuron. These electrical chemical signals happen from 5 to 50 times per second on every neuron, and these travel through specific though very complex pathways, forming the neural network. Officially labeled as 3,2-aminoethyl 1H-indole 5-ol, serotonin has a variety of other common names, some of them with historical importance, and others associated with this neuroreceptor nomenclature. In the structural formula, it can be observed the presence of the functional groups of an aromatic alcohol, a primary and secondary amines. These are some of serotonin's physical properties. In the brainstem, serotonin is produced in a region called Raphe nuclei. It is synthesized from the amino acid tryptophan, which besides its protein importance, it also is the precursor for other essential molecules such as melatonin and vitamin B3. The production of serotonin happens in two main steps. First, a hydroxy group is added to carbon 5 of the indole ring, catalyzed by the enzyme tryptophan hydroxylase. This generates the 5-HTP molecule, which is also widely sold and consumed as a dietary supplement because it can cross the blood-brain barrier, where serotonin itself cannot. This molecule then loses its carboxyl group in a reaction catalyzed by 5-hydroxytryptophan decarboxylase. This generates the serotonin, or 5-HT molecule, which is then consumed and used up in all of its processes. After being utilized, serotonin is finally metabolized through substitution of its side chain primary amine for a carboxyl group. This generates 5-hydroxyindoleacetic acid, which is the final metabolite of serotonin. This is then eliminated in the body through urine. The story of serotonin begins in the early 1930s at the University of Comparative Anatomy and Physiology, University of Pavia in Italy. Vittorio Sparma had a devotion to the study of extracting pharmacologically active substances from live animals, a field that was called pharmacognosy. Dr. Sparma was interested in the smooth muscle constricting and contracting properties of various amine substances found in the skins and intestinal tracts of species including rabbits, mollusks, and frogs. One substance which interested him a lot was found in enterochromaffin cells of the gut, an acetone isolate of the cells called smooth muscle contraction. He then deduced that the substance was an epinephrine, and that on the basis of color tests, it was likely an indole. In 1937, he named the substance enteramine. Another very important researcher was Arvin Page, the director at the Cleveland Clinic. Dr. Page was specialized in questions of hypertension and atherosclerosis. Through his research, he believed that hypertension could best be explained by the presence of endogenous constricting factors in the blood. However, whenever blood was studied for such a factor, another substance was produced as soon as the blood coagulated. Dr. Page realized that this serum substance would have to be removed from the blood preparations before any progress could be made on the hypertension producing factor. For this, Dr. Page recruited a highly skilled organic chemistry, Morris Report, and a highly skilled biochemist, Ardald and Green. This team, in 1948, finally succeeded in isolating and characterizing the serum substance, and they named it serotonin, which literally meant serum that causes constriction. The isolation process was achieved through an acetone precipitation to remove other solutes and the methanol extraction, which resulted in the thin, rhomboid pale yellow crystals. A year later, Dr. Maurice Report determined the structure to be 5-hydroxytryptamine. Dr. Report was collaborating with synthetic chemists at the Upjohn Pharmaceutical and Abbott Laboratories until when in 1951 they finally made available synthetic serotonin for research. Another crucial researcher was Betty McTwalrog, who was investigating the retractor muscle of the mollusk Mytilus. Betty had identified the contracting neurotransmitter to be acetylcholine, but she couldn't identify the relaxing one. Norepinephrine was weakly active, but it was evident that a new yet unidentified neurotransmitter was involved, which had chemical similarities to both epinephrine and the ergot alkaloids. When Betty saw the studies from the Cleveland Clinic on serotonin, she was very interested because the structure fulfilled the structural requirements she had established for the Mytilus neurotransmitter. Shortly after, when serotonin and enteramine were identified as the same substance, Dr. Twarog finally realized that the unknown neurotransmitter was indeed serotonin. Dr. Twarog was also convinced that invertebrate neurotransmitters could also be neurotransmitters in vertebrates. So, working with a clam-hard preparation, which he had refined to be extremely sensitive, Dr. Twarog finally found serotonin in mammalian brain. This discovery ultimately brought serotonin into the field of neuroscience. Lastly, Dr. D.W. Woolley combined the work of Betty Twarog, showing serotonin existing in the brain, with Albert Hoffman's discovery of LSD and his own work on LSD. In spite of his blindness, it is usually said that Dr. Woolley saw the structure of serotonin in LSD and realized that LSD could be used as an anti-metabolite tool to uncover the role of serotonin in mental illnesses. Finally, this opened up a whole new potential for the treatment of mental health issues to this day. Serotonin has evolved to be a mediator of an animal's perception of resources availability. In less complex animals, such as invertebrates, this simply means food, and its abundance then is correlated with high levels of serotonin, while low levels would then indicate scarcity, consequently also signaling higher levels of stress. In more complex animals, though, resources can also mean social dominance, meaning that individuals who are higher in the social hierarchy would also have higher levels of serotonin. This abundance or scarcity of serotonin related to the subjective perception of resources can further signal growth and reproduction, which can then be triggered or put on standby, and ultimately regulate the animal's overall mood. Recent studies also show that dominant and confident posture and body language are also associated with higher serotonin levels and indicate better moods overall, while the typical depressed posture, with shoulders and spine hunched forward and the head tilted down, correlate with low levels. Serotonin then, in this way, affects not only the neurochemistry of mood, but also its physiology. In most mammals, serotonin is responsible for the vastest neural network, modulating and regulating a variety of different functions. In association with dopamine, it is present in the reward circuitry. While dopamine gives the motivation necessary for completing a task, serotonin is associated with the feeling good sensations that come afterwards, like when some important activity is completed, after a good meal, and after sexual activity, to name a few. The majority of its neural circuitry, though, is found in what is called the social brain, usually associated with oxytocin. While oxytocin is related to the creation of social bonds and trust, serotonin is the main motivator to seek social interactions, being released upon perceptions of social acceptance and social dominance, as was mentioned earlier. Recent studies are also relating serotonin to cognitive processes, being found in the hippocampus that's associated with memory processing and is related to prefrontal cortex information processing speed. Details of most of these mechanisms, though, are still not very understood. Serotonin has been historically associated with psychedelics, since most of the compounds that cause altered states of consciousness and can trigger these hallucinogenic experiences act upon the serotonergic 5-HT neuroreceptors, some of which, mainly the tryptamines like DMT and psilocybin, commonly known as magic mushroom, also closely resemble the structure of serotonin itself, as is shown in more detail in the next slide. Another mild hallucinogen, MDMA, which is the active compound of Molly and ecstasy, directly releases serotonin from the synaptic vesicles. Some of the more potent psychedelics, like the synthetic LSD and the naturally occurring DMT, famous for its ayahuasca tea from Amazon natives and the psilocybin mushrooms, if used under proper supervision and while in elevated states of mind, can trigger a rush of elevated emotions of gratitude for life and nature. And users usually report experiences of expansion of awareness and consciousness and feelings of oneness with the cosmos. This has been seen as increasingly potential of treatment for depression and mental health issues. The brain mechanisms underlying these experiences are still unclear, but it has been correlated with an increase in the activity of the serotonin receptors of the thalamus, the major responsible for sensory input into the cortex, thus creating these altered sensory experiences, which are typical of the psychedelic experience. The vast majority of prescribable antidepressants are somehow related to serotonin, as it is now understood how it is closely associated with mood elevation. One type of these, the monoamine oxidase inhibitor, prevents the oxidation of monoamines, such as serotonin, dopamine, and epinephrine, ultimately increasing its availability in the brain. The serotonin norepinephrine reuptake inhibitors prevents the reabsorption of these into the presynaptic cell, while the selective serotonin reuptake inhibitor performs the same role on serotonin only. This ultimately causes an increase in extracellular levels of the neurotransmitter available for synaptic activity and binding, which increases the overall activity of the serotonergic pathways. Although widely used, especially in the US, these prescribed antidepressants do have a potential for dependency, and it is known that they yield better results when used as an addition to cognitive behavioral therapy. Thank you, everyone, and have a great summer.